Alright guys, this is Matt Dean with A Plus College Ready, and our lesson today is chi-square testing. Chi-square is a statistical test used to compare a set of observed data to an expected data distribution. The test can be used to accept, or as statisticians prefer us to say, fail to reject a null hypothesis, or to reject the same null hypothesis. The null hypothesis is essentially the idea that two groups of data, observed and expected, are not statistically different, or that the independent variable in your experiment had no effect on the outcome. For example, if you wanted to state a null hypothesis for an experiment in which you're using fertilizer and measuring its effect on plant growth, your null hypothesis might be fertilizer has no effect on plant growth. If you're simply comparing two data sets, your null hypothesis might be there are no statistically significant differences between the observed and expected data sets. So this is a piece of the AP Biology formula sheet, and this is the part that contains the chi-square formulas. And if you look closely, this looks pretty intimidating. Chi is a Greek letter that looks like a curvy X, so chi-square equals, here's sigma. Sigma means summation, and it's the summation of the quantity O minus E squared all over E. And that's where O stands for observed and E expected. Also in this section of the formula sheet is this data table right here, and this table is called the critical value table or the chi-square table. From this table we'll obtain a critical value that will be critical for us to compare to our chi-square statistic. We'll talk more about that table in just a minute. So here's what an example problem might look like for chi-square. The M&M company claims that its bags contain 30% red, 20% brown, 10% yellow, 20% green, and 20% orange candies. Now realize that the Mars company does publish the percentages that should be in the bags. These are not the real numbers. These are numbers that I made up. But these numbers are what we're going to call our expected numbers. And then a little bit further down, it says after opening the bags and actually counting the contents, the following data were obtained. So when we actually counted the M&Ms in the bag, we got 50 red, 60 brown, 30 yellow, 40 green, and 20 orange. So the first thing we want to do is state a null hypothesis. And our null hypothesis here, since we're simply comparing two sets of data, is that there are no statistically significant important differences between our expected data and our observed data. And we're going to perform a chi-square analysis and determine if that chi-square is true or not. Are the sets of data close enough that the small differences are due to chance? So let's go to the next slide and actually calculate a chi-square statistic. Alright, so whenever we're calculating a chi-square statistic, the first thing that we want to do is make a chart. And we want the chart to have the labels that you see up here. Phenotype, O stands for observed, E expected, O minus E quantity squared, and O minus E quantity squared all over E. So the phenotypes are the possible outcomes. In this particular example, those would be our colors of M&Ms. So our colors were red, we'll just write R for red, brown, yellow, green, and orange. And if we went back and looked at the stem of our question, it told us that when we actually counted the M&Ms, that we actually counted 50 red. The actual data, those are our observed data points. It also said that we counted 60 brown, 30 yellow, 40 green, and 20 orange M&Ms. Now to get the expected data set, that's where we're going to use those percentages that the M&M company told us that we ought to get. But before we can use those percentages, we need to add up the total number of M&Ms in our sample. So there's 150 plus 60 is 110, plus 30 is 140, plus 40 more is 180, plus 20 makes 200 total M&Ms. So to get our actual expected values, we're going to take the percentage that M&M said we should get of each color and multiply it by our total number of M&Ms, which is 200. For example, for red, it told us that 30% of the sample should be red. So we're going to take 30% written as a decimal, 0.30, and we're going to multiply that by our total number of M&Ms, 200. When we take 30% of 200, we expected to get 60 reds. We're going to continue that with brown. We expected to get 20% brown M&Ms. 20% of 200, 0.20 times 200 is 40. For the yellows, we expected to get 10%. 10% of 200 is 20. For the greens, 20%, which would give us 40 again. And for the oranges, also 20%, which gives us 40 again. All right, so now to finish the chi-square calculation, we'll just do what the column labels tell us to do. So the next column is observed minus expected. So for the red row, we'll go observed 50 minus expected 60, and we'll get minus 10. For the brown row, 60 minus 40, 20. For the yellow row, 30 minus 20. The green row, 40 minus 40. And the orange row, 20 minus 40. In the next column, we're just going to square O minus E. So for the red row, we've got to square negative 10. Negative 10 squared is positive 100. Remember, when you square a negative, it becomes positive. 20 squared is going to be positive 400. 10 squared, positive 100. 0 is just 0. And negative 20 squared is 400. Final column is O minus E squared all over E. So we're going to take, for the red row, 100, O minus E squared, and we're going to divide it by E, 60. Now you could do this in a fraction, but it's probably easier just to go ahead and do 100 divided by 60, and that gives you 1.67. For the brown row, O minus E squared, 400, divided by E, 40, you get 10. The yellow row, O minus E squared is 100, divided by E, 20, so you get 5. Green row, O minus E squared is just 0. And then for the orange row, O minus E squared is 400, divided by 40, E, that gives you 10. So remember that the chi-square statistic is equal to the summation of O minus E squared all over E. So what that tells us to do is simply to add up this last column to get our chi-square statistic. So if we add up that whole last column, 10 plus 5 plus 10 plus 1.67, we get a chi-square statistic of, let's see, that would be 26, and I want to round it off to the 10th place, 26.7. Alright, so we just calculated a chi-square statistic of 26.7. By itself, that doesn't really tell us anything. We have to compare that to the critical value, which comes from this table down here. Before we can get a critical value, we need to know how to use our chart. First thing you need to know is what are called degrees of freedom. When you're doing a chi-square in this class, the degrees of freedom are going to be your number of phenotypes, or possible outcomes, minus 1. So in this particular problem, we had 5 colors of M&Ms. Those would be our phenotypes, or possible outcomes. We're going to take away one of those. 5 minus 1, we have 4 degrees of freedom. Also, we have to know the proper p-value to use. For AP Bio, unless you're told differently, you're always going to use the 0.05 level. That's essentially almost like an error level, and I'll explain more about that in a minute. So if we use 4 degrees of freedom and a p-value of 0.05, we get a critical value right here of 9.49. I'm just going to abbreviate that as CV. All right, so we're comparing our critical value of 9.49 to our chi-square statistic of 26.7. And in this case, our chi-square statistic is higher than our critical value. If that is true, if the chi-square statistic is higher than the critical value, that means that you have to reject the null hypothesis. Again, you do that if your chi-square statistic is higher than your critical value. And what that means is that our null hypothesis was that our observed data and our expected data weren't significantly different. This is telling us that they are. That the M&Ms that we counted did not follow that pattern that we thought we should get. So we're going to reject the null hypothesis. And again, you do that whenever your critical value is less than your chi-square statistic. All right, this is Matt Dean with A+ College Ready again. We're going to look at our second chi-square example problem. In this case, we're going to look at one that deals with genetics, in particular a fruit fly cross. So the problem says, in the P generation, a female fruit fly, homozygous for white eyes, is crossed with a wild-type male. Wild-type in this case means red eyes, and this male is homozygous for red eyes. All of the F1 male flies have white eyes, and all of the F1 females have red eyes. So the F1s are the children of the P generation flies. Out of 100 F2 offspring, the following offspring were obtained. So notice in the F2, the generation that's the children of the F1s, we got some red-eyed females, some red-eyed males, some white-eyed females, and some white-eyed males. So our first question is, what is the most likely mode of inheritance for the white eye color trait, and why? And then draw a Punnett square that depicts the cross that produced the F1 flies. So first of all, let's figure out what's going on here. So in the P generation, we had females that had, all the females had white eyes. So I'm just going to use a W there for white. And the males all were wild type, red eyes. In the F1, all the females ended up with red eyes, and all the males ended up with white. And then in the F2, we got a little bit of each. So we need to figure out how that worked. And really the key to figuring this out in terms of the mode of inheritance is to look at this offspring right here. The first clue for a mode of inheritance should be that at least between the P generation and the F1, males and females seem to be affected by this trait in different ways. Whenever that's the case, a good bet about your mode of inheritance is that it has something to do with the sex chromosomes, that it's sex-linked. We know this white eye color trait can't be Y-linked because females are getting it sometimes. So if it's sex-linked and it's not Y-linked, that tells us that this trait is most likely an X-linked trait. We need to know as well if it's a dominant or a recessive thing. That's where we want to look at this offspring. This female got one of her two X's from dad and the other from mom. So she now has one red carrying X and one white carrying X, and she has red eyes. So that tells us that red is a dominant trait. Therefore, white must be X-linked and recessive. And red is X-linked and dominant. This guy, this F1 male, he got his only X from mom. He got a Y from dad. So therefore, that makes sense that he has white eyes. So let's write out some genotypes here. So in our P generation, our female, which we need to show our sex chromosome since we're talking about X-linked traits, our female was homozygous for white eyes, which we now know is recessive. We're going to show that as a superscript, a little r, because it's recessive. Our P generation male, he's XY. We're going to show his big R because he was dominant for red eyes. We don't put anything on the Y chromosome in terms of a superscript because it's an X-linked trait. So one of the first questions was to show the Punnett square that produced the F1s. To make the F1s, we need to cross the P generation. So if we were to draw a Punnett square for that, our female can make gametes carrying an X chromosome with a white allele on it. Our male, on the other hand, can make gametes with either the red carrying X or the Y. So now when we fill in this Punnett square, these are the genotypes we get. The phenotypes would be, these two are both female, they're XX, and they both have a big R, which means they're red-eyed females. On the other hand, these have an X and a Y, and their X only has a little r, which means these are all white-eyed males. So that explains why in the F1 generation, all the females had red eyes, all the males had white. Now the next piece of information, or the next question, is to show a Punnett square explaining how we got the F2 generation. So again, let's make a little space. We'll draw that Punnett square right here. So to get the F2 generation, we need to cross the F1 individuals. So in the F1, all our females had this genotype, and all our males had this genotype. So the females can make eggs that either carry a red X or a white one. In the males, all the X's are carrying the white allele, and the Y's aren't carrying either one. Which means we can get these outcomes. Now what I hope you see here is that this offspring is female and has a big R, which means this is a red-eyed female. This offspring is XY, so it's a male, and it has a big R, which means it's red-eyed. This offspring is female, XX. Both of its alleles are little r's, which means it's a white-eyed female. And this one is a male, XY, has a little r, which means it's white-eyed. So we're expecting 1/4 red-eyed females, 1/4 red-eyed males, 1/4 white-eyed females, and 1/4 red-eyed male or white-eyed males in the F2 generation. Those are our expected numbers. In a genetics kind of chi-square problem, we're going to get at least our expected ratios from a Punnett square. These numbers, these are our actual observed numbers. These are the offspring that we got when we actually did the cross. So our chi-square statistics, or our chi-square test, the job of that test is to compare observed data here to the expected ratios to see if the differences are small enough that they're probably simply due to chance. So let's run a test. Let's do a chi-square test on this data. Alright, just like we did in the M&M problem earlier, we first of all need to figure out our phenotypes. So in this case, our phenotypes are going to have to do with eye color and also gender. Anytime you're doing a chi-square problem like this and it turns out that it's linked to the sex chromosomes, you need to write your phenotypes in terms of both trait and gender. If we determined that this trait was autosomal, we would only need to write down red and white. Gender wouldn't really matter. But in this case, since we're dealing with an X-linked trait, color and gender both matter. So I'm going to notate this or shorthand it. I'm going to write R for red. So we're going to have red eyes, and I'm going to put M for male, red-eyed male. White-eyed male, red-eyed female, and white-eyed female. Right, now our observed numbers are what we actually got when we did the cross. So we got those from the stem of the problem. And the stem of the problem told us that there were 26 red-eyed males, 26 white-eyed males, 23 red-eyed females, and 25 white-eyed females. So now you may be wondering, how do we get the expected values? Anytime you want to get the expected values, the first thing that you want to do is add up the total number of data, the total of your data set. In this case, the total number of flies that we observed. So if we add up 26, 26, 23, and 25, those add up to be 100. Now to get the expected numbers, remember that our F2 Punnett square a minute ago told us that we should get 1/4 this, 1/4 this, 1/4 this, and 1/4 this. So we're going to take 1/4 of 100 to get the expected numbers for each one of these. 1/4 of 100 is 25. So we're expecting 25 of each phenotype. From here on out, we just fill in the table, just like normal. So we're going to go O minus E, 26 minus 25, 26 minus 25, 23 minus 25, and 25 minus 25. Now we're going to go O minus E squared. So for the RM column, the red-eyed male column, that would be 1 squared. For the white-eyed male column, again, that would be 1 squared. For the red-eyed female column, it would be -2 squared. Remember, when you square a negative, you get a positive. And then this one would be 0. Finally, O minus E squared all over E. So for the red-eyed male column, that would be 1/25. Again, it's probably easier to write this as a decimal, so 1 divided by 25 is 0.04. It'll be the same on the next column, or the next row, 1/25, 0.04. The next one is going to be 4 here, over 25 here. 4/25 is 0.16. And then this one's going to be 0. So our chi-square statistic, remember that chi-square is the summation of O minus E quantity squared over E. And that tells us once again to simply take this last column right here and sum it up. So when we do, we end up with a chi-square statistic of 0.24. Now again, by itself, that doesn't mean anything. We need to compare this chi-square statistic to our critical value. To get that critical value, we have to go back to our chi-square chart. Alright, so remember that our chi-square statistic was 0.24. We need to compare that to our critical value, which we'll get from our chart down here, our chi-square table. In this particular setup, we had 4 possible phenotypes: red-eyed males, white-eyed males, red-eyed females, and white-eyed females. So we had 4 possible outcomes, 4 phenotypes. Remember that for degrees of freedom, we're going to take our number of phenotypes or possibilities minus 1. So 4 minus 1 gives us 3 degrees of freedom. And unless we're told otherwise, we're going to use this 0.05 p-value, which means we have a critical value in this case of 7.82. And we need to compare chi-square here, 0.24, to critical value here, 7.82. In this case, our chi-square value is less than our critical value. And what that tells us is that our observed data, the flies that we counted, the numbers that we counted in each category, that those were very close to what we expected. Therefore, we're going to fail to reject the null hypothesis. Some people would say accept the null hypothesis. But again, statisticians want us to say fail to reject. What that means about our data is that our data, the differences between the observed data and the expected data were very, very small. And more than likely, those differences are just due to chance. So again, when chi-square is less than the critical value, you're going to fail to reject the null hypothesis. And this tells us that our observed data is near our expected data and that the differences are small and probably due to chance. So let's look back at the rest of the questions and make sure that we clearly stated everything. For a problem like this one, our null hypothesis would simply be that our observed number of flies is not statistically different from our expected number of flies in each category. We constructed the chi-square analysis table. We got a chi-square value of 0.24. So our chi-square value, once we worked it out, was 0.24. Our degrees of freedom was our number of types of flies, or phenotypes, minus 1. So we had 4 groups, minus 1 gave us a degrees of freedom of 3. Our critical value from our chart was 7.82. And since chi-square was less than the critical value, that told us that we're going to fail to reject the null hypothesis, which told us that the flies that we observed, in terms of their numbers, those numbers were very close to what we expected, and that the small differences were probably due to chance. So one more time, when your chi-square value is lower than your critical value, you're going to fail to reject your null hypothesis. All right, this is Matt Dean with A+ College Ready, and this is our third and final chi-square example problem. In this particular problem, a researcher found that onion root tip cells spent approximately 90% of their life cycle in interphase and 10% in mitosis. He then treated an experimental group of onion root tip cells with lectin, it's a chemical, and found that after being treated with lectin, 700 of the onion root tip cells were in interphase and 1,600 were in mitosis. So the first thing we're asked to do here is to state a null hypothesis. In this case, we're doing an experiment. Null means none, nothing. So what we want to say in our null hypothesis is that essentially whatever our independent variable is, that it doesn't have an effect. So in this case, we're going to say that lectin has no effect on the cell cycle of onion root tip cells. That would be a valid null hypothesis. It may not be true, but essentially it's saying that the experimental treatment has no effect. Again, our null hypothesis for this particular problem is going to be that lectin has no effect on the cell cycle of onion root tip cells. So we're next asked to perform a chi-square analysis on this data. So this one is going to be much shorter than the problems we've done in the past. For phenotypes, we're only going to include 2 here. Our cells are either in interphase, which we'll abbreviate with I, or they're in one of the stages of mitosis. So those are the only 2 phenotypes we're going to use, interphase and mitosis. Our observed data, what we actually got when we counted the cells, the problem stem told us there were 700 cells in interphase and 1,600 in mitosis. Now again, what bothers people is how do you get these expected numbers? The stem of the problem told us that normal cells not treated with lectin, 90% of the cells were in interphase and 10% were in mitosis. So we're going to add up the total number of observed cells. 700 plus 1,600 get 2,300. To get those expected values, we're going to take the percentages from our question stem. 90% of cells are in interphase, in cells not treated with lectin. So we're going to say 0.90, we're going to convert it to a decimal, times our total number of cells, 2,300. And when we do that, we're expecting 2,070 cells to be in interphase. To get the number of mitosis expected cells, we're going to take 2,300 times 0.10, 10%, and that tells us that we expect 230 cells to be in mitosis. To finish chi-square, we're just going to follow our column labels. So O minus E, 700 minus 2,070, we're going to get negative 1,370. In the mitosis row, 1600 minus 230, we'll get positive 1370. All right, then we have to square O minus E squared. So that gives us 1,876,900. And that comes out to be the same here, 1,876,900. Finally, O minus E squared over E. So we'll take the 1,876,900 and we're going to divide that by the expected value for interphase, which is 2070. When we do that, we get 906.71. We'll do the same thing for the mitosis, 1,876,900 divided by 230, and that gives us 8160.4. It's supposed to be. All right, so once we add up that final column, we get a chi-square statistic of 9,067. I'm going to round it off to the 10th place, 0.1. Keep in mind that the AP exam math problems will tell you where to round off your values to. Round them to the tenths, round them to the hundredths. In this case, I'm going to round off to the tenths. Again, this number by itself doesn't tell us much. We have to compare it to a critical value. So we have to go back to our chi-square or p-value table. So again, our chi-square statistic was 9,067.1. We need to compare that to a critical value from our table. In this experiment, we only had two phenotypes, two outcomes, and that was either interphase or mitosis. So our degrees of freedom are our number of outcomes minus 1, 2 minus 1. We have 1 degree of freedom, and we're going to use the 0.05 p-value, which puts us at a critical value of 3.84. We compare chi-square to the critical value. Chi-square is much higher than the critical value. When that's true, remember, when chi-square is higher than the critical value, we reject the null hypothesis. Remember that our null hypothesis was that there was no statistically important difference between our observed and expected data. This is saying, when we reject the null hypothesis, that there are differences. So summarizing, since our chi-square statistic was higher than our critical value, we're going to reject the null hypothesis, and this tells us that our expected values and our observed values were very, very different, and that those differences were too large to be simply due to chance. Now I want to go back and address this 0.05 value here. This tells us that, in this case, even though we're rejecting the null hypothesis, there's still a 5% chance that maybe we shouldn't have. That's what the 0.05 deals with. It's like an error level. There's a 5, within 5% is good enough for biological purposes. Hope that helps. Let's go back and take a quick look at the question stem and make sure we answered all the parts of the question. All right, so again, the first thing that they asked us to do in this problem was to state a null hypothesis. And since we were comparing or looking at the effect of lectin, we said that lectin has no effect on the cell cycle in onion root tip cells. We did our chi-square. We found that our chi-square statistic was much higher than our critical value. That told us to reject the null hypothesis. What that means in this case is that lectin apparently does have an effect. Because our expected values were very, very different than our observed values. So what our test here is telling us, lectin is affecting the cell cycle.